Hello, I will talk about glycogen metabolism today. After a short review of the structure and function of glycogen and the importance of glycogen storage and glycogen metabolism in different cells, I will talk about the glycogenolytic pathway and the glycogen synthetic pathway, which we also call glycogenesis. Then I will talk about how we can regulate that pathway and also a side note about glycogen storage diseases that are related to the lack of one or the other important enzyme in the process. Again, the Word file, which is covering the same material, is posted on Entrada in the Basic Metabolic Pathways folder, and there are two short videos I found on YouTube that you can potentially use to review the material. So, where is the glycogen synthetic and degradation pathway in the whole big metabolism? This is, you remember, this is glucose glycolysis, gluconeogenesis, and when glucose is coming into the cells, can also be used for glycogen synthesis to store away glucose for further use. And through glycogenolysis, glucose phosphates can be liberated from the stored glycogen and can be pushed to the glycolytic pathway for energy, or in the liver can be pushed to release glucose, which will help to balance the circulating blood glucose level in our body. So what is glycogen? Glycogen is polyglucose. Every little dot stands for a glucose molecule. You may recall that this is the so-called amylose chain when the glucose units bind to each other through alpha-1 4 glycosidic bond. And this is building a long chain in the glycogen. However, there are branch points in which a glucose unit binds to another one with alpha-1 6 glycosidic bond. And from this junction point, another amylose chains can start to build. Another straight chain can start to build. This machinery, which is building or degrading glycogen, is located in the cytosol. Every cell of ours can store away more or less glycogen for further use. So the tissue, all the tissues, there are two tissues that store away more than the others, and it is liver and muscle. And it is a cytosolic process, so really anaerobic, aerobic cells can do this, can save away glucose, and then they can liberate glucose phosphate from that. So important in the liver and the muscle. It is 10% of wet weight of liver can be glycogen. 1 to 2% of the wet weight of muscle can be glycogen. If you add all this together, which tissue contains more total glycogen? Yes, the muscle, because we have more muscle than liver. So, glycogenolysis is breakdown of glycogen to glucose phosphate. Glycogenesis is synthesis of glycogen from glucose. There is a difference between liver and muscle in the use of glycogen. They both can synthesize glycogen in the same way, but the use is different. In muscle, the glycogen is turned into glucose 6-phosphate and channeled to glycolysis and serves as a store-away energy carrying molecule. In the liver, the glycogen is turned into glucose 6-phosphate, but it is not going to glycolysis. It is actually going to the glucose 6-phosphate enzyme system in the ER, if you remember, and will produce glucose. So that is the enzyme system that produces free glucose, not phosphorylated glucose in the liver, either through glycogen degradation or from gluconeogenesis, which will play a role in balancing blood glucose. So why cannot muscles supply blood glucose? Because it doesn't have glucose 6-phosphatase in the cells. This is something that I brought here to demonstrate that we actually use liver glycogen a lot. So liver glycogen is there to balance blood glucose. So let's say this is the morning. You have a little leftover glycogen in your liver, but you eat breakfast. Please eat breakfast. If you eat breakfast, which has carbohydrates in it, then it will be stored away in the form of glycogen. By the time you hit lunchtime, you use this glycogen up pretty much because you need to do a lot of things that will drop your blood glucose level low, and then the glycogen will be released. Glucose will be released by liver to balance it. So glycogen store will be depleted. Now, depending on how much glycogen you started with, depending on what you do, whether you just sleep from 8 a.m. to noon or you actually come to class and study hard and running the marathon in the same time, then you will use a lot of glycogen there. All right, now you eat a little lunch. Unfortunately, in this country, lunch is not in the center of our attention, so you build back liver glycogen a little bit, which you will happily use up by dinner time. And then the big dinner comes and glycogen is built up. But as you see here, by breakfast time, you use up your liver glycogen and you need to eat your next breakfast to do that. It is different when you starve. Of course, there are no lunches, there are no dinners, there are no breakfasts. So this particular liver glycogen can supply blood glucose for a day or less, again, depending on what you are doing, if you are not eating in between. So this is to demonstrate where and is glycogen, where to look for, and how it looks like. So this is electron microscopic picture of a liver cell, an animal liver cell, and you see this beautiful flower-like, it says chrysanthemum-like, little things in the cytosol. These are all collagen blots or dots or big molecules. So we have glycogen not only in one little spot in the cytosol, but we have glycogen in many clusters here in cytosol. Now, if you look at not electron microscope, but just simple microscopic picture, you see the cells and you see the glycogen stained with red. This counter stain is, the purple is for nuclei. So you see that in the cytosol, a lot of glycogen can be stored away in a well-fed animal. Now, this is to compare a fasted and a fed animal. Now, the brighter pink, purplish pink is glycogen, and this blue is nuclei, of course. So glycogen is actually almost depleted completely in a fasted, only overnight fasted animal. You can see a little remainder in the cells of the liver, but when you feed the animal in the morning, you see that glycogen is building up pretty nicely. So, what do we use muscle glycogen for? We use it for energy during muscle activity. This is an easy way of gaining energy. You may recall that we talked about creatine phosphate, which is also sitting in the muscle cytoplasm, and this molecule preserves as much as 1 ATP worth of energy, and it can be done with very easy hydrolysis by the CK enzyme. So that's number one, really within a second you can get the ATP from there. But muscle glycogen liberation of glucose phosphate, putting them into glycolysis can also generate ATP pretty quickly. Now, what is the difference between red and white muscle cells? Red muscle cells, blood, and white muscle cells. Red muscle cells, blood, mitochondria, everything. So glucose 6-phosphate, when channeled into glycolysis, the pyruvate will go to mitochondria and lots of energy can be produced. You remember 38 ATPs from one single glucose molecule. And when glycogen is going through glycogenolysis, not only one molecule, but hundreds or even thousands of molecules of glucose can be produced very quickly. On the contrary, white muscle cells that have poor blood supply and very few, if any, mitochondria, they do anaerobic glycolysis, which means that from one glucose molecule coming from glycogen, only 2 ATPs can be produced. So white muscle cells usually store away much more glycogen than red muscle cells do to have enough quick energy available to support the working of the white muscle cells. Now, it also means that glycogenolysis, the tapering down of glycogen, is happening very fast and a lot of energy will be used or liberated. So it means that the white muscle cell can exert itself, but only for a short period of time. So for comparison, chicken breast muscle is white and the heart muscle is red. So chicken cannot fly because the white muscle doesn't have enough energy for a long flight. If you are looking at the muscle of the flying birds, that red muscle cell, that can use glycogen to start with, but then it continues to use fatty acids to produce a lot of energy and also for a long time. All right, again, structure of the glycogen, just to repeat, what I wanted to show here is that we need the branches to occur because the enzyme which is building the glycogen is building at the non-reducing end. And the enzyme which is tapering down the glucose phosphate molecules also starts at the non-reducing end. So the first reducing end is the starting glucose unit, and from there amylose chain is built, which then builds a junction point, a branch point where another amylose chain can start, another branch point, other amylose, and another amylose, branch. It is calculated that branches occur approximately every fourth glucosyl unit. So you can imagine how dense that particular glycogen is. So this non-reducing end can accept glucose, and then this non-reducing end will be from where glucose phosphate is liberated. So why do we need the branch? To be able to put away more than one glucose at a time, because one amylose chain would be able to accept only one glucose, and also liberate lots of glucose molecules from all of these non-reducing at the same time. Now, why do we store glucose in glycogen? Why not fat? So I said already that it can be mobilized fast, while the fat to be used for energy takes longer time because the fatty acids need to be released from the adipose tissue, needs to be delivered to the target tissue, needs to be taken up by the mitochondria, should be channeled to the beta oxidation, then the Krebs cycle, then terminal oxidation, so it takes much more time. Also, glycogen can be utilized in the cytosol without oxygen, so every cell can do it, while fatty acid utilization is only possible in aerobic cells with mitochondria and oxygen. Glycogen can be converted to glucose, fat cannot be converted to glucose, except for that tiny little bit of propionyl coenzyme A from the out-chain fatty acid. Why don't we store it in glycogen and not glucose? Glucose can go to any cell, you remember? There are the GLUT transporters there. However, glucose is osmotically active and it brings in water. Glycogen is not. So this is a comparison that to replace with glucose 0.01 micromolar of glycogen, we would need 400 millimolar of glucose in the cells. Impossible. All right, let's see glycogen degradation, which we call glycogenolysis. The key enzyme is called glycogen phosphorylase. Please remember that. Glycogen phosphorylase works on the non-reducing ends of the amylose chains. All right, what does it do? This is not simple hydrolysis. Do you remember digestion of polysaccharide in the digestive tract by amylase? That is a hydrolase that will take a starch or glycogen, which are very similar in structure, and they will hydrolase off free glucose from the non-reducing end. Now, glycogen phosphorylase, however, uses a molecule of inorganic phosphate and produces glucose 1-phosphate out of the glycogen. It also acts on the non-reducing ends, but the end product is phosphorylated glucose. Glucose 1-phosphate will be mutated to glucose 6-phosphate, so this is a mutase enzyme. And then in the liver, you remember, glucose 6-phosphatase can turn it to glucose. In non-liver, glucose 6-phosphate will channeled into glycolysis, anaerobic cells, lactate, aerobic cells, complete oxidation will happen. Why is it good that we have glucose phosphate and not simple glucose? Because glucose 1-phosphate can easily turn to glucose 6 and directly go to glycolysis. Now, this enzyme is working on amylose chain until we reach the branch points. If there would not be an other enzyme that will help us, then we would just be able to taper off a couple of glucose molecules until we reach the branch point, and then phosphorylase would stop there and we would be doomed. The glycogen cannot be fully utilized. Now, one more point. Phosphorylase needs vitamin B6. So B6 is important, pyridoxal phosphate, to be taken up with. Now, as I said, if phosphorylase would work, it would stop before the branch points. So phosphorylase is working until that on that particular amylose chain, we have 4 glucose units left. Steric hydrons, phosphorylase cannot work more. So, luckily, we have an enzyme which is very logically called deep branching enzyme, which has 2 activities. One is a so-called transferase activity, when it takes 3 of the 4 glucose units and transfers it to the end of another amylose chain. So you see it's here. And we are left with 1 glucose unit. This 1 glucose unit is hydrolyzed off, taken up of the junction point and produces glucose. And this is the debranching enzyme glucosidase activity. So transferase activity and glucosidase activity needs to work, but this is the same enzyme with 2 active centers. So we have glucose 1-phosphate, a lot, and we have a free glucose. Is this good? That depends where we are. So if we are in the liver, yeah, that's good, because we are producing glucose anyhow, and it can go out of the liver cell and go to the blood. However, when we are, for example, in the muscle, this glucose needs to be phosphorylated with 1 ATP to glucose 6-phosphate, so we are losing one very precious ATP to activate this glucose to go to glycolysis. So this is the summary. Glucose, glycogen phosphorylase, glucose 1-phosphate, glucose 6-phosphate goes to glycolysis depending on anaerobic or aerobic. We produce energy from that glucose unit in the glycogen. If we are in the liver, glucose 6-phosphatase will produce glucose. And here is the reminder of where this particular enzyme sits. So, as I mentioned, there might be some problems with these enzymes, which will result in so-called glycogen storage diseases. They are not particularly very frequent, 1 in 20,000, 1 in 25,000 births, but it's worth mentioning because there are a couple of subtypes that result in pretty serious consequences. So let's start from type 2. I will talk about type 1 on the next slide. So type 2 is so-called Pompe's disease. You probably remember that lysosomes are the recycling center of the cell. Glycogen, especially when it is a lot in the cytosol, can go to the lysosome and it needs to be degraded there. There is no phosphorylase there. There is a glucosidase, a hydrolase enzyme there, which works in an acidic pH. So if this glycogen, which goes there, cannot be degraded because this glucosidase enzyme is deficient, there will be serious consequences: liver, heart, muscle, cardiomegaly. It can even lead to early death. Why? Because if something is going to the lysosome, it's not able to be degraded, it accumulates there, leaves the lysosome, kills the lysosome, kills the cell, damages the organ. All right. Type 3, Cori disease. This is the lack of the debrancher. So phosphorylase is okay. It actually tapers down the amylose chain, but it needs to stop 4 units before the branch point. It results in fasting hypoglycemia. Why? Because there is no debrancher, which means that amylose can be tapered down, but it stops. Amylose tapered down, stop, 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 stop. So we only can use the glucose or glucose phosphate from the periphery of the glycogen. It will stop at the first branch point from the outside. So why fasting hypoglycemia? Because we cannot produce enough glucose in the liver in Cori disease. Now, it also says that the glycogen has abnormal structure. Why? Because the synthesis can go on. So the synthesis will build more amylose chains, more branch points, longer amylose chains, another branch point, and we only can taper down the periphery. So there will be a lot of glycogen in the cytosol of these patients, of the cells, and it will not be reduced substantially. So liver, heart, muscle may be getting some damage. Definitely mild fasting hypoglycemia is the sign of Cori's disease. Now, 5 and 6. 5 is so-called McArdle, skeletal muscle specific, and type 6 is Hirsch, and this is liver specific. Same disorder, phosphorylase deficiency, but this is only in the skeletal muscle and this is only in the liver. So the consequences are the same. If there is no phosphorylase, we cannot liberate energy from glycogen stored glucose. So that's the problem, because this particular quick energy source is not available. So cramping of the skeletal muscle, even skeletal muscle damage can happen. So myoglobinuria means that the myoglobin, which is an oxygen storage protein in the cytosol, will leave the cell. CK3, LDH5, aldolase, myoglobin will be detected in the blood, which all relates to muscle cell damage. All right, high level of glycogen, of course, because phosphorylase is not working efficiently. So this is the same in the liver. Phosphorylase is not there. Glycogen is growing, but it is not reduced in size. And fasting hypoglycemia, which is the sign. Similar to CORES, glycogen is not available to produce glucose for the circulation. So gluconeogenesis need to work harder to produce glucose. Finally, the most serious is type 1, which is so-called von Gürk's disease. 1 in 100,000, but it is very serious. So I mentioned before, and you may remember, that this is the problem with the lost enzyme system, which is glucose 6-phosphatase. Why enzyme system? Because either the translocase that puts glucose 6-phosphate across the membrane to the ER, or the enzyme is not working well, you will not have glucose produced. So 1A is the enzyme deficiency, 1B is the translocase deficiency. It affects liver, and because kidney also has this enzyme and participates, especially in starvation, to release glucose, liver and kidney will be affected majorly. So von Gürk's inability to produce glucose. So there will be hypoglycemia, of course, fatty liver, hepatorenal progressive renal disease, growth retardation, delayed puberty, lactic acidemia, hyperlipidemia, hyperuricemia, and so on. Very serious metabolic derangement because of the lack of this single enzyme or the translocase. So there is no problem with the muscle. There is no problem anywhere because they don't produce glucose anyhow. Okay. So hepatorenal fatty acids, renal disease, hypoglycemia, which is severe now. Why is it severe? You remember? This enzyme is the last enzyme in both glycogenolysis and gluconeogenesis. No glucose comes from the liver. Very bad. Lactic acidosis, hyperuricemia, hyperlipidemia. I give you a little bit of explanation of that one. So hypoglycemia, obvious, no glucose. Lactic acidosis, because the hormones are signaling for glycogen to release glucose phosphate. It goes to glycolytic pathway, and after the glycolytic pathway, it goes to lactate. The gluconeogenesis is also inhibited because of the lack of enzyme. So pyruvate is accumulated, and something needs to be done with the pyruvate. So it turns to lactate. Pyruvate also can go to the TCA, can be used for fatty acid synthesis. Hyperuricemia, because we need glucose and we need ribose 5-phosphate from glucose to produce nucleotides. And we have a lot, and this is pushing the purine and pyrimidine synthesis, so they will be overproduced. Purines, because they are extra, they will degrade, and it can lead to gout. So we are under glucagon signal Glycogen degradation is a must. Gluconeogenesis would be upregulated, but because of this enzyme deficiency or translocase deficiency, neither of them can go and produce glucose. This is a typical picture of a baby, big stomach, chubby face, and very thin extremities. All right, now very quickly, glycogen synthesis. How do we start? Chicken or the egg? So as I said, this is the only reducing end of the very first glucose. All the others are non-reducing ends. This reducing end is actually how the whole thing starts, that there is a specific protein, not just one, many copies of this protein, which we call glycogen, in the cytosol. It has a tyrosine residue, which the first glucose, through its active reducing hydroxyl, will bind to. So this protein is glucosylated with the first glucose, and then the first glucose can react with the second, third, fourth, whatever, and the whole glycogen network can build out around this tiny little protein. And because we have many copies of this protein in the cytosol, many of these glycogen clusters can be built. All right. Now, how do we synthesize? Then glycogen. What do we do after the first glucose is bound to glycogen? We need to activate the glucose. You remember, every biomolecule that we want to enter into a pathway needs to be activated. So we activate through hexokinase, glucose 6-phosphate will be produced. A phosphoglucomutase, exactly the same as it worked in the reverse direction in glycogenolysis, will mutate glucose 6-phosphate to glucose 1. And then there is the specific enzyme for glycogen synthesis, which we call glucose 1-phosphate uridine transferase, which will further activate the glucose unit. So this is the UDP glucose, which is finally active and can enter into the synthetic process. So this will be the UDP glucose. And when we go further, this UDP glucose will be reacting with every end, one molecule with every non-reducing end glucose residue in the glycogen. So the glucose N in glycogen will grow with 1 and UDP will be liberated. The enzyme which is putting the glucose on the non-reducing end is called glycogen synthase, key enzyme. Please remember, glycogen synthase, again, can only build amylose chains. So we need an extra enzyme. Extra enzyme which will build the branches. We call it branching enzyme. So how does this work? It works in a way that when you have 7 residues, 1, 2, 3, 4, 5, 6, 7 residues of glucose synthesized here, then this branching enzyme takes off the 7 glucose units, so it was added here, and informs a branching point, putting the glucose into an alpha-1,6 linkage. So amylose grows, 7 units taken off by the branching enzyme and put into a branch point. Summary: glucose 6-phosphate, activation step 1, it's the first step of glycolysis also. Glucose 1-phosphate activated UDP glucose, and UDP glucose is actually building the synthase is building with that UDP glucose amylose chain, and the branching enzyme will put the branch point there. How do we regulate this very important system? When does the glycogen goes and produces glucose phosphate or glucose in the liver, and when will the glycogen grow? We need to regulate the key enzyme glycogen phosphorylase in the glycogenolytic process and glycogen synthase in the glycogenic process. Allosteric regulators and hormones will act, as we talked about it in the glycolytic and gluconeogenic process. So let's see at first glycogen phosphorylase. When do we need to degrade glycogen? When we are in the low glucose level. When do we need that? When we need glucose. So, glucose 6-phosphate, when it is enough in the cytosol, will inhibit glycogen degradation because we already have enough. ATP, high energy level, will also inhibit because we have enough energy. There is no need to produce more molecules that can go to glycolysis to produce more energy. And glucose itself will inhibit the process. So this is in the liver, very logical. What happens in the muscle? Glucose 6-phosphate, ATP, is inhibiting the process. Glucose is not. But look at that, there is a positive allosteric regulator, which is AMP, which will turn on glycogenolysis and glycolysis, and calcium. So when the muscle contracts, needs more energy, then glycogen degradation will start. How do we do the hormonal regulation? Now, bear with me. This looks like a very complicated picture, but it's not as complicated as you think. So watch out for this. When an enzyme is labeled A, that is active. When the enzyme is labeled with P, it is inactive. Now we already know that we need to degrade glycogen when there is need for energy or when there is need for glucose in the liver. So epinephrine and glucagon will signal. Both of them go through G protein coupled receptors. Both of them will upregulate cyclic AMP protein kinase A, and this protein kinase A will phosphorylate a so-called glycogen phosphorylase kinase. So originally it is inactive in its B form, and when protein kinase phosphorylates, it is in the A form. So the phosphorylated form is active. And this is a key enzyme to phosphorylate and activate glycogen phosphorylase. And this is the active form, phosphorylated, and it's A, and then it will start glycogen degradation. Glucagon, epinephrine effect will be reversed by insulin. When insulin is signaling to the cell, it means that there is plenty of glucose, so there is no need to upregulate glycogen degradation. So insulin will dephosphorylate phosphorylase, turning into inactive, and it will also dephosphorylate the glycogen phosphorylase kinase, turning it inactive, so glycogen degradation stops. Now, there is a note here that calcium is acting in these enzymes, and glycogen is actually calcium in the muscle. Calcium is turning this enzyme without phosphorylation to the active conformation. And then it can phosphorylate glycogen phosphorylase. So this is the role of these allosteric regulators that can actually circumvent the hormonal signal, and when there is a need for glycogen degradation, allosteric effectors can do that. All right, so regulation of the synthase. You remember opposing pathways need to be regulated in the opposite way. So glucose 6-phosphate downregulates phosphorylase, but it upregulates glycogen synthase. It's logical because when glucose is coming into the cells, and it is both for muscle and liver, then glucose 6-phosphate will be produced, and when the concentration of the glucose 6-phosphate is high, then it will turn on the synthase and saves away some glucose in the form of glycogen. Now, hormonal regulation is very similar, but of course it will be the opposite. When glucagon and epinephrine is signaling and protein kinase is cyclic AMP and protein kinase is activated, then it will, of course, phosphorylate glycogen synthase, but in this case, the phosphorylated form is inactive. Is it not great? With the same hormone signal, we upregulate the glycogenolytic process, and in the same time, we are inhibiting the synthetic process. Now, don't worry about this particular figure. I just wanted to show you that it's not the only kinase that can do the job, but other kinases too, including phosphorylase kinase, protein kinase A, of course it was labeled here, phosphorylase kinase, which phosphorylates phosphorylates synthase, upregulates phosphorylate, downregulates synthase. Okay? Now, I put something here just to think this whole system through. Putting together now glycogen and glucose, this is what you can see. First of all, I have a question. How do you know that this is liver, as I put into the title, and it is not skeletal muscle? Guess. Right. So you have glucose secreted to the blood, which means that glucose 6-phosphatase is here, so it cannot be anything else but liver and not muscle. Tiny little bit of kidney. Okay, so glucagon epinephrine effect on liver glycogen metabolism. We just went through that. Epinephrine, glucagon, adenylate cyclase, phosphorylation, which will activate glycogen degradation, phosphorylase, and it will inactivate glycogen synthesis. Now, cyclic AMP also acts on inhibiting glycolysis because the phosphorylation of glycolytic enzymes, majorly, by the way, kinase in glycolysis, will inhibit glycolysis. Why is it important? Because we want glucose to be leaving the cell. We don't want to put this into energy generation or fat synthesis or anything else. We want glycogen to supply glucose. Okay. Go further. Now you know it is muscle, correct? Because it is not glucose, but it is either lactate or carbon dioxide produced by the glycolytic process. So what is the hormone? Muscle is not sensitive to glucagon. It is sensitive to epinephrine. The same thing, beta-adrenergic receptor, adenylate cyclase, protein kinase, phosphorylase activates it, phosphorylate synthase inactivates it. But you remember in the muscle, by epinephrine signal, glycolysis will be upregulated at the PFK1 level. So, why is it good? Because when glucose phosphate is coming from glycogen, it is channeled right away to glycolysis and it produces energy for the cell. Now, calcium, as I mentioned before, upregulates glycogen degradation, and it is through the acetylcholine receptor, the nerve impulse comes. Calcium is released from the endoplasmic reticulum, which will lead to the same events as we mentioned before. Opposite, insulin. First, I am asking this question, which one the above figures is liver and which one is muscle? Hopefully you did see the solution. GLUT4. GLUT4 is only in the muscle, not in the liver. Okay, so glucose is coming in, goes down to hexokinase, glucose 6-phosphate is produced, glucose 1-phosphate goes for glycogen synthesis. So when insulin signals, then it will dephosphorylate phosphorylase, which stops glycogenolysis, and it will dephosphorylate synthase, which will lead to upregulating the synthetic pathway. And in the meantime, you remember insulin signal is needed to put GLUT4 from cytosolic storage to the membrane to take up more glucose. And the liver is just doing insulin dephosphorylation events. Degradation of glycogen is inhibited, synthesis of glycogen is upregulated. Now, this is the same that I showed you in gluconeogenesis. Please read it again. Very important to remember that newborns, infants, children, and especially premature and small for gestational age infants are different from adults. They need to be fed with carbohydrates, with glucose, to keep them healthy and growing. And this is a summary figure: glycogen synthase, glycogen phosphorylase, regulatory events. This is the synthesis and degradation, what occurs in muscle, cytosol, UTP is needed. Well-fed state means insulin. Fasting state means glucagon and epinephrine. Thank you. I forgot about summary. Stored form of glucose. Liver, muscle contains a lot. They are different because liver is using it for balancing blood glucose, muscle is using it, and every other cell uses glycogen to produce energy. Phosphorylase and synthase are the two key enzymes, remember the names. Allosteric effectors and hormonal regulation both plays role in making a determination whether glycogen needs to be degraded or glycogen can be synthesized. Please remember again, glucagon epinephrine kinases, phosphorylation, stimulating phosphorylate, inhibiting synthase. Insulin, phosphatases, dephosphorylation, stimulate synthase and inhibit phosphorylase. And enzyme deficiencies lead to glycogen storage diseases. Thank you.